Recall that there are two types of cellular transport, passive and active. Passive requires no added energy, but active does require added energy. Active transport also moves from an area of low concentration to an area of high concentration, or up the concentration gradient. This is the opposite of passive transport, and because it's more difficult, it needs the energy molecule called ATP, which stands for adenosine triphosphate, because there's an adenosine molecule and 3 phosphate molecules. There are 3 types of active transport: membrane pumps, endocytosis, and exocytosis. Let's look at membrane pumps first. Membrane pumps are just carrier proteins that move substances from areas of low concentration to areas of high concentration. Let's look at the sodium-potassium pump to see how it works. Outside the cell, there's a high concentration of sodium ion, or Na+. The brackets represent the word concentration. Inside the cell, there's a high concentration of potassium ion, or K+. The cell has ion channels that will allow these ions to diffuse and reach equilibrium. But the cell doesn't actually want that. It wants to keep a difference in concentration. So the sodium-potassium pump will force more sodium out and more potassium in all the time. Let's see how this works. First, 3 sodium ions from the cytosol bind to the protein, and a phosphate group from ATP breaks off in the carrier protein. This provides energy for the shape of the protein to change, and the sodium ions can be released into the outside of the cell. Now 2 potassium ions will enter the pump, which causes the phosphate to break off and return the pump to its original shape and release the potassium into the cytosol. Again, sodium will diffuse into the cell on its own, and potassium will diffuse out. The pump forces the sodium to be outside the cell and the potassium to be inside the cell. Another way that cells do active transport is through vesicle movement, either endocytosis or exocytosis. Endocytosis brings items into the cell, and exocytosis pushes them out of the cell. Endocytosis is the process by which cells ingest external fluid, macromolecules, or other large particles, even other cells. We can be more specific about what materials undergo endocytosis. Phagocytosis is where large particles or whole cells are ingested, and penocytosis is when solutes or fluids are ingested. You can think of phagocytosis as cell eating and penocytosis as cell drinking. Here you can see the way it works. First, the cell membrane begins to pinch around the substances, and eventually it forms a vesicle which can carry the substance to any part of the cell. Exocytosis is basically the opposite of endocytosis. It's a process by which a substance is released from a cell through a vesicle that transports it to the cell surface and fuses with the cell membrane. A vesicle can merge with the cell membrane because they're both made of phospholipids. The layers press into each other, and the phospholipids rearrange a little so that they can open up their contents to the outside of the cell. Again, this requires energy, so it's classified as one of the types of active transport. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter @SciencePet.